Let's take a closer look at the structure and function of the RNA molecules that participate in the final stage of information transfer. Most RNA molecules are single-stranded, though they often contain regions of secondary structure. They are long linear molecules, that is, they are unbranched, and contain the same type of phosphodiester bonds as in DNA. The major distinction between DNA and RNA is that the 5-carbon sugar or pentose is beta-D-ribose in RNA rather than deoxyribose. Another difference is in the type of pyrimidine bases, including uracil and cytosine, but not thymine. As we consider the structure of tRNA shown here, we immediately recognize the presence of secondary structure. Nevertheless, it is still a single-stranded RNA molecule with one 5' phosphate end and one 3' hydroxyl end. This RNA molecule is responsible for carrying the amino acid to the ribosome via an ester bond at its 3' OH. The black lines joining the red segments represent regions of intramolecular complementarity and are hydrogen bonding interactions. Thus far, we can see that tRNA exhibits both primary and secondary structures. The native form of tRNA contains tertiary structure as well, as pictured on the left. The loops in the central figure marked 1 and 3 contact one another to form the L-shaped tertiary fold. Some of the bases in these loops have been chemically modified, as shown in the structures on the far right. The actual synthetic machinery is that of the ribosomes, composed of two differently sized subunits. The bacterial subunits are shown here and include the smaller 30S and the larger 50S subunit. These subunits contain both protein and rRNA, but are primarily composed of RNA. Though the mass of the subunits varies between prokaryotes and eukaryotes, they each have large and small subunits. Analytical ultracentrifugation may be used to monitor the dissociation and reassociation of ribosomes. In this instrument, particles move within a density gradient based on size, with larger particles sedimenting more readily. The quantitative measure of sedimentation is the Svedberg coefficient, symbolized by the capital letter S. The larger this value, the heavier the subunit. As we examine the structure of ribosomal RNA, we find it too has secondary and tertiary structure. Similar to tRNA, there are regions of interchain complementarity that result in canonical base pair formation. Notice the extremely large and detailed structure of rRNA on the right. As in the case of tRNA, this complex molecule folds into a stable, compact structure with a distinct tertiary fold, pictured on the left. The message that carries the genetic code, mRNA, is synthesized in its original form as heterogeneous nuclear RNA or hnRNA. This contains the introns to be spliced out, mentioned in the previous lesson. mRNA molecules have a limited lifetime in the cell and are present in relatively small amounts. The mRNA molecule that is eventually translated into protein was first synthesized from information contained within the DNA. The process of transcription involves separation of the DNA strands to produce an RNA complement to one of the strands. With the complexity shown in these RNA molecules, we can already see that they must have vital and specific roles to play in information transfer.